All right, so the first thing we're going to talk about is just kind of an overview of the immune system, very brief, very quick. And then we're going to talk about a little bit of the physical and the chemical barriers. I'm going to roll through those quite fast, as you've seen them before, with a couple of exceptions. And I really want to get us to a couple of processes that we're going to draw out, which is primarily how inflammation works in the body, what it's there for, and how it combats all of these pathogens that we get in. So first, we're going to talk about the innate versus the adaptive immune system, and the basic differences between those. This is a really good point where you should get like a blank piece of paper and make you a T-chart, because you will have to know the different components of the innate versus the adaptive system. Big theme that we're going to talk about. Another big theme that we're going to talk about is how we protect things that are outside of cells versus things that are inside of cells. All right, so it's a good another T-chart. And we can refer to these as extracellular intracellular. And I would just have these two little T-charts nearby. You can just start adding things to them. All right, so our innate is our first line. This is what's known as the immediate defense. And these are just things that are always there helping protect you from bacteria. Simple as your skin and the oils on your skin. So we'll go through all of these examples. This begins before birth. It's always active. So immediately when a pathogen hits, it is active and it's ready to go. While the infection and the disease persists, it's still working. All right, so this is always on. The adaptive immune system is a little bit more specific. So adaptive immunity takes time, it's delayed. And by delayed, I mean anywhere from 3 days to a week in many cases before it actually kicks in. But it's called adaptive because it actually learns. So with the innate immune system, bacteria bad, let's get rid of it. That's pretty much the logic behind it. The adaptive immune system is going to learn about that bacteria that's infecting you. It's going to take notes, it's going to find its weakness, and it's going to make specific weapons to that bacteria so that it is really, really effective at clearing it out, which takes time. It just takes time to collect information, to make whatever weapons you need to make, and then mount a response against that. The adaptive is also what protects you from recurrent infections. And we'll go through all the details about all of this stuff. Today, we're primarily just going to be focusing on this innate column over here, though. So let's run through a couple of physical and chemical barriers. So first and foremost, most obvious physical barrier is going to be our skin. And it's more than just our skin is there and it's keratinized cells and it's waterproof and things can't get through it very easily. It's all the other parts of the skin that we've talked about as well. We have oils on our skin. These oils contain fatty acids, which lower the pH on our skin. Our skin can be salty. And again, our skin is selective media. The bacteria that can grow on our skin, we want to be on our skin most of the time. All those Staphylococcus species that we've seen, their presence just in and of itself is a protective layer on our skin. They prevent other things from colonizing. We have flushing mechanisms of our skin that we've talked about. So sweat glands not only provide moisture and salt on the skin, it also acts as a flushing mechanism to get rid of things. Our outer layer of epithelial cells on our skin will shed after they mature, so you get sloughing off of the skin cells. So this is to prevent, especially viral infections from lingering and hanging around. We talked about this a lot when we were talking about HPV and how it actually has to get into the basal cells that are cut before it can infect the cells. And then actually takes advantage of this process in order to spread. One thing we haven't talked about yet are these alt tissues. All right, so we're going to see lots of these salt, malt, and galt. And these are associated lymphoid tissues, all right? Just like you see here. These are built-in immune arrays that kind of exist inside of your body. All right, we have one on the skin. We have one in your mucous membranes. And these are essentially like little sentry towers on your body. They're always on the lookout for something that's not supposed to be there. And when something that's not supposed to be there is observed, these will start an immune response. Specifically on the salt, excuse me. These are a specialized cell known as a Langerhans cell. Langerhans cells. This is a popular term that you see if you're reading about immunology and inflammation. So these are essentially white blood cells that live in the skin. It's really right below your skin more than anything. And we'll talk about their function when it's time to. Along with your skin, you have mucous membranes. So we talked about mucous membranes before. A very, very large surface area of your body, much more so than your skin in terms of what's being exposed to the outside area and pathogens. So this is the inside of your lungs, which are constantly breathing, and the inside of the digestive tract. So all the food and other things that get swallowed all go through these mucous membranes. Not only do they act like big fly traps, kind of hold everything down, they can move the mucus around. So we've talked about the mucociliary escalator in your lungs, how it can move bacteria and dust and foreign particles out of your lungs and up into the back of your throat, where you can cough it up or even end up swallowing it. On these mucous membranes, we all have more microbes that live there, so especially when we're talking about the gut microbiome, and their presence, just like on the skin, is a protective presence. It holds the fort down, doesn't let anything else establish itself very easily. And here is our mucosa-associated lymphoid tissue, and then we actually separate out the gut, even though it's part of the mucous membrane system, because of how important this is. So GALT is a part of the MALT system. And when we talk about the gut tissues, there's really one little area that we really care about in this class, and that is, how does your gut determine what bacteria are supposed to be there, so the good ones, and which ones are the bad ones? We've referenced this issue several times back when we were talking about microbiomes, but we weren't ready to really learn about how it works yet. So inside of your intestines, you have these epithelial cells that surround it. Those are the tight junction cells that keep things from leaking out. It's not supposed to be there. Well, within those cells, you have these microfold cells, which are often just labeled M cells. It stands for microfold cells. And these are essentially little sampling mechanisms. So your microfold cells, your M cells, They live in this, I can't spell today. Live inside of the epithelial layer, so it's just like the surrounding. And these are associated with lots of different white blood cells. A specific type called a dendritic cell, which I don't know about yet, but you will. And these white blood cells have little arms, and they'll peek through inside of the gut. So this is inside the gut. And sample little bacteria cells that it finds. And they'll grab onto one and pull it down and just check it out. So it's kind of like these little arms that reach into your intestines. They grab bacteria cells and they pull them down, and they interrogate them to see if they were supposed to be there or not. And if they're not supposed to be there, we start an immune response. If they are just normal gut bacteria, they just kind of ignore them and move on and keep going. Right, so this is our system to kind of know when we have a bad bacteria, like a Salmonella cell, versus a good bacteria, like an E. coli cell that's supposed to be there. And these exist in Peyer's patches. So here's the inside of an intestine. You can see all the villi here. Here are our little follicles where contain the M cells. And here are our little dendritic cells that live inside. These are macrophages in this case. And they just sample all kinds of things that come through here. Your lungs are a little different than your gut. There's not as much bacteria that lives there, even though there are some. So they have a little bit more of a passive way of protecting them, I guess you could call it. So inside of your alveoli, so the deepest little crevices of your lungs, we just have some white blood cells that kind of live in there and hang out. And you don't know what a macrophage is yet. Those are like the stereotypical white blood cell you see in cartoons that go around eating other cells. That's all a macrophage is. And these just kind of hang out there. And if something happens to pop up that deep in your lungs, it's more of a sensor. So it's like a, hey, ding, something's here. Let's figure out what's going on. If it's made it that far into your lungs, though, it's probably set off lots and lots of other alarms well before it set off these alarms. Along with that, we have our mucociliary escalator that we all know about. And then, of course, bulk expulsion, which is our coughs and our sneezes, which get lots and lots of things out at the same time without having to worry about how slow this little escalator works. Right, again, we've seen most of those already, with the exception of the associated lymphoid tissue like malts and galts. But those are pretty straightforward in what they do, and you'll understand them even better once we get through some more of these concepts. Chemical barriers, again, you've seen all of these before. We've talked about them. We have stomach acid, very, very acidic. Very few things can live in there. What does live in your stomach tends to live in the mucus that lines your stomach more so than actually in your stomach. We have lysozymes. So we talked about lysozymes in your tears, in your eyeballs. These are little enzymes that break down cell walls. They're particularly effective against gram-positive bacteria and also exist in mucus. We have reactive oxygen species. So that's what this superoxide radical refers to. We just replace that with reactive oxygen species, or ROS. We know how damaging that is. So what our cells do is we make a little organelle. We fill it up with reactive oxygen species. When we find a bacteria, we shove it into that little pocket full of reactive oxygen species and let it destroy it for us. Right? Those are called peroxisomes. So you can think of that like hydrogen peroxide, peroxisomes. We'll see these again in the future as well. One that we haven't really talked about, these things called defensins. And these are basically just little protein-like things. It's a big category of molecules, so we're not going to go into detail about it. we have lots of other molecules that we don't have time to talk about, and we're basically just gonna classify all of those into the defensin box. All right, so. Are there questions about chemical or physical barriers? I know I went through those pretty quick, but we've seen them several times before.
So what do we need to know about defenses?
Defensins are just the other stuff that exists that we don't have time to cover. That's all defensins are gonna be for us. Because there's lots and lots of different types of them.
So you just need to know the name, pretty much?
Yeah, just defensins refer to the other miscellaneous things that we haven't specified. All right, so let's actually start looking at some new stuff. So primarily, where does our immune system reside in our body when it comes to the more specialized cell types? So the physical and the chemical barriers are great, but we want to look at all of the white blood cells that actually circulate around and eat up that bacteria. So our organs, we can classify into primary organs, or lymphoid organs, and secondary lymphoid organs. Primary is easy because it's just the two. We just have bone marrow and our thymus. So these are where white blood cells go to mature. You can write this down. We're not going to really go into detail about these until Thursday. Secondary is a little more all-encompassing, so the rest of your lymphoid tissue is pretty much your secondary. The main ones are going to be your lymph nodes. So everybody's heard of lymph nodes before, probably in the context that they swell up when you get sick. So lymph nodes are where we transfer information from infections. All right, so when your white blood cells go out and find bacteria, lymph nodes are kind of like the little hubs where everything converges and your immune system gets activated and learns about all of this stuff. Right, so they're super, super important in activation. Some other things, your spleen. So your spleen is actually part of your immune system as well. Your spleen's primary function is filtering out a bunch of red blood cells that are dead or dying. But it also has lots of white blood cells in it. They're looking for diseases that happen or pathogens that might be hiding in your blood somewhere. Your tonsils and your adenoids. So these are all around the top of your throat, the top of your digestive tract. These are basically like little screening centers. So your tonsillar crypts, whenever you swallow food or things that go through your mouth, they kind of act like a little catch and will trap particles inside of your tonsillar crypts where they're exposed to your immune system. So it kind of, it's like a little filter where it's screening what's going through and trying to detect any anomalies, any pathogens that could be in there. And that's mainly for things that go in this way. Other ones could be your appendix. Your appendix is one that for a long time thought had no purpose. Now we kind of know that it actually houses a bunch of beneficial bacteria and can allow a repopulation in the event of a die-off. There are some remnants of old secondary organs that exist in our kidneys. They're called tube cells. For those of you who have had a nephrology background in A&P, may have learned about tube cells before. And those used to be the equivalent of our tonsils, but for the other end. But we've kind of moved past that and don't need that anymore with our gut microbiome in the way. Here's where things are, tonsils and adenoids up top. Here's your appendix down here at the bottom. Peyer's patches inside of your intestines. Lymph nodes tend to aggregate in the joints. So the insides of your arms, your armpits, the groin area, behind your knees. This also tends to be where you get sore from certain types of immunizations. So like the flu shots and the COVID shots. Some people would get really, really sore right here on the inside of their elbow or right under their arm. And it almost feels like a pulled muscle, but it was actually swollen lymph nodes from there. Your tonsils can swell up if you get something like strep throat, and you'll see that. So those are a type of lymphoid tissue. And that's lots and lots of cells converging in those locations and being very active. So that's why these places tend to be the ones that either hurt or swell up during certain types of sicknesses or illnesses. Now, inside of these places are where all of the cells kind of interact. And we want to learn about these different cell types here. These are leukocytes. I know you've heard these terms before. Leukocyte is just a general term for white blood cells. Lymphocytes is a specific type of white blood cell that we'll talk about on Thursday. But in any case, we're talking about white blood cells. They circulate in your body. Many of them live in these lymph organs, specifically these secondary lymph organs. And we often classify, at least the ones that we're about to talk about, as poly, morpho, nuclear lymphocytes, or leukocytes actually. And that is a super long name that essentially just means many shapes, nuclei. And oftentimes it's called PMN cells for short. These are named as such because whenever we stain their nuclei, they all have really weird shapes. Usually in most of our cells, if you look at a nucleus, it's just a little sphere in the middle. It looks almost like a mini cell inside of a cell, just like every diagram you've ever seen in a eukaryotic cell. But these guys have lobed nuclei, so these are known as lobes. So for example, this is a neutrophil, this is a eosinophil, and you can see that the nuclei look odd. And then all around here are red blood cells. So all of these red discs that you see are the red blood cells. There's a white blood cell, another type of white blood cell. So quite easy to identify in a blood smear, which is what you're seeing here, based off of their size as well as the lobing that you see in the nucleus there. And this is usually how we identify what type they are, is by what the lobing looks like. Not that you will have to do that on your own. Neutrophils are kind of like the main one. When you hear white blood cells, you're usually only talking about one of two, which is neutrophils, which is the ones that are in your blood, and they're circulating around. They're super abundant. There are tens of billions of these in your blood at any time. They're circulating around and waiting to be needed, essentially. The other one is that macrophage that I was talking about, the little Pac-Man cell that goes around and eats bacteria. Neutrophils can also eat bacteria, so that's our phagocytosis there, if you haven't forgotten that term. However, these often use those reactive oxygen species to kill the bacteria they eat. So they ingest a bacteria, they shove it down into an organelle that's full of reactive oxygen species, and it breaks it up. Those reactive oxygen species are also toxic to us. So one of the problems with neutrophils is when you get a bunch in an area, they start releasing all of that reactive oxygen species or byproducts of that reaction, and it's actually toxic to us as well. So we have to kind of be careful our balance here when we're using neutrophils. These are easily the most abundant white blood cells in the body that we monitor quite frequently. The next two are basophils and eosinophils. These are often noted in their use in allergic reactions. So we see a lot of eosinophil, basophil activity in allergic reactions. There can be other things that kind of trigger them too. Eosinophils can also be triggered by parasitic infections. We're not going to talk about these too much. These are mainly like chemical mediators, so they don't necessarily go out and eat other cells. They kind of sit still wherever they're at, and if a bacteria or a virus is around, they just kind of like set off a horn and an alarm. And they signal all the other cells to do the work, but they kind of huddle up and hang tight and just yell directions around. The mast cell is similar to this. It's a little bit more prominent in examples that we're going to be using today, like in your skin. These also just kind of hang tight inside of your skin in that interstitial layer right underneath your dermal layers. And they are the ones that really kick off an immune response. I always call these the war horns, because as soon as these get a signal that there is a foreign entity in your body, they just dump chemicals that say, danger, danger, activate the immune system. So in almost all the cases in the examples we use, it's the mast cells that are the major alarm system inside of the body. A couple of other types. We'll actually start at the bottom here. Macrophages and dendritic cells. So these are the macrophages, so little Pac-Man cells. They're the ones that go around and really eat a bunch of other cells, including dead neutrophils. So as neutrophils do their job and they die, macrophages will actually scoop up all those toxic byproducts so they don't damage our tissue and then transport them away. Dendritic cells are very similar to macrophages. Dendritic cells are a little bit more specific though. So macrophages kind of go around as a cleanup crew. Dendritic cells connect wherever you get the infection to your lymph nodes. And let's see if we can make some notes of this instead of. All right, so we have like a site of infection. And then we have your bloodstream, your lymph system, so all your lymphatic vessels. And then we have nodes, lymph nodes. So this is really connected by your macrophages. So they're out circulating around. We get some type of infection, let's call it on your hand, for example. They're going to go over to your hand. They're going to clean up whatever your neutrophils have destroyed, and then they're going to go into your lymph system and just drain out, essentially. And we'll see how this this whole process looks together. So we'll make a big diagram at the end. Your dendritic cells have similar capabilities. Dendritic cells, except they go to the site of the infection, collect those bacteria, and then take them over to your lymph nodes. We will explain this more as we go on, so it's okay if you don't get this right away. I'm just going to start building the foundation of what's going on here. So if we look at the functions of all of these different ones, we can actually get an idea into what is attacking the body. So you probably heard of a differential panel with white blood cells before, at least some of you have. So white blood cell differential, we actually look at the counts of total white blood cells and then each of these individual ones that we see down here. And when one of them, I didn't tell you what a monocyte was, did I? Monocytes are just precursors that will turn into one or the other as needed. So monocytes, they just hang out, and it's like, oh, we need more dendritic cells, they will turn into dendritic cells. If the body is like, hey, we need more macrophages, then they will turn into macrophages. So they're kind of like the on-call precursor to fill in the staffing of wherever we need more cells and the kinds of cells. But back to our panel, we can look at which ones are raised, which ones are lowered, find patterns, and kind of have a guess into what's going on in the body. For example, viral infections, we usually see an increase of lymphocytes. For parasitic infections, we usually see an increase of eosinophils. For allergies, for their allergies, we usually see an increase in basophils. This can also be eosinophil disorders as well. This is not the end-all be-all of a diagnosis, mind you. Just knowing what is being increased doesn't necessarily confirm a diagnosis. It just gives you an idea of what's going on inside of the body. All right, any questions on those cell types as of right now? All right, so we've talked about these being the Pac-Man cells and utilizing reactive oxygen species as a weapon against bacteria. So let's get a little bit of a review on how phagocytosis works with a little bit of extra information as well. So if you remember, phagocytosis is the endocytosis of another cell. In this case, it's a foreign cell or foreign body. So we have the white blood cells. These are the good guys. And then we have some type of bacterial cell. But we need to destroy that bacterial cell. So how do we actually do that? Just like in endocytosis, except more active, we will ingest this by wrapping around it and get it inside of the cell. That's our first step. This is a lysosome. And then since we already have our pocket full of ROS here, so these are peroxisomes, or what we really want to do is fuse these two. So we will direct this over and fuse them together. So now our result is the bacteria cell is exposed to all of this ROS. And the fusion is often called a phagolysosome, which is the fusion. All of this ROS will essentially break down, kill that cell, break it down into lots of little bitty pieces. And then all of those pieces get ejected out. So this is primarily, we're looking at, we're going to call this a neutrophil, just for the sake of this drawing. So neutrophil gets to the site of the infection. We find some bacteria cells, we eat those cells, we digest those cells after we kill them, and then we can spit out all the pieces of the cells. So the cells are no longer a threat to us. However, that also releases some of these toxic byproducts. So you kind of have this old battlefield where all your neutrophils have gone in and killed those bacteria. And there's just debris and chemicals that exist in that area afterwards. So if we want to clean those up, we would have a macrophage come through. They would clean up. We're going to see this in action in a big process. Building pieces here. Sometimes these guys like to be able to slip out and not get captured. Anybody tell me some defensive measures that these take to prevent being phagocytosed? A capsule is probably the best example. So let's say this guy has a capsule. And that capsule prevents us from capturing it. Capsule. All right, so he tries to grab it, just slips away. Tries to grab it, slips away. How can we bypass that capsule? That is what this process of opsonization is. All right, so this is, instead of just trying to grab this slippery capsule, why don't we just coat it in a bunch of antibodies and make it rough, right? So it's like having a slip surface and then covering it with sand instead so that you don't trip. So what we can do is, hey, we have antibodies. Let's cover it in those antibodies. And it's a whole lot easier to grab onto. So coat pathogen in antibodies. AD just stands for antibodies, so that it isn't slippery. So we just have a way to facilitate this process if the bacteria has a defense against it. Here is opsonization here. Here's bacteria. If we coat it in these little antibodies that we see here, it makes it very, very easy for us to latch onto it now. In fact, antibodies have an area that specifically sticks to our white blood cells, so it makes it even more than just sticky. It makes it want to bind and be captured by our cells. So quite an efficient way to improve the process of phagocytosis, especially in your blood or in your skin. Some things don't care if they get eaten by your white blood cells. In fact, they want to be eaten by your white blood cells. So the best example I like to use is Salmonella here. Salmonella will actually willingly get taken up into your cells and then prevent it from ever fusing with that ROS, so that they can just kind of live in your cells until they make it into your bloodstream. Shigella can do very similar things. Shigella and Salmonella are very closely related. Listeria is another one that will purposefully get ingested into your white blood cells, and then it will break out of that little lysosome that it's caught in, and then just happily live inside of your cells. All right, so this is how Listeria gets in there. Coxiella, if you remember Coxiella burnetii, it's the one that we test that pasteurization is designed to kill. This one actually doesn't even care if it gets into contact with the ROS. It can survive right through that. It has lots of antioxidant capabilities. So this is not a surefire way of killing these cells for those that have at least evolved their defense around it. Now, the big question is, this is us and our immune system. How do we know that this is something we need to attack? What happens if our immune system attacks our cells? What's that called? Yeah, that's autoimmunity. We don't want that. We only want our immune system to target the bad cells. So how do we know which cells are bad and which cells are us, or self cells, as they would call it? And that has to do with what I call the cellular barcodes. So all of our cells essentially have a barcode on the outside of them that our other cells can scan. And this is the major histocompatibility complex proteins. They're MHC. These are essentially your immune system genes, at least in a sense of the word. And it's a combination of proteins on the outside of your cell. Here's our cell. So our cell is coated with lots of proteins that stick out. And some of these are to signal hormones, to signal other cells. Lots and lots of different signals and receptors on here. One set of them, though, is your MHCs. These are MHCs. And your immune system can go on and scan it. So your immune cell comes up, it scans this, it says, okay, that is the correct MHC. You don't need to attack this cell at all. Your MHC is completely unique to you unless you have an identical twin. Right? The only person who has the same exact MHC as you would be your identical twin. Other people can have very similar ones, but the chances of you having the same MHC as anyone else in this state or even in this country is quite low. They're very, very unique. You get half of it from your mom, you get half of it from your dad, which is why these are the genes that are used in maternal testing, because they're so unique to you and so unique to a father that the chances of those matching and them not being directly related as an offspring are next to zero. So very, very unique to you, which makes it good at determining which cells belong to you and which cells belong to somebody else. In what cases might you have a different MHC in your body?
Stem cell infusions.
A what?
Stem cell infusions or blood infusions.
Infusions,
Transplants.
transplants, one more. What? Pregnancy. Pregnancy can also introduce another one. We'll talk about the effects of that. That's what rejections are when it comes to transplants. Now, bacteria don't have anything this complex in their systems. So if we're looking at bacteria cells or even viral cells, these will have very simple patterns on the outside of them. And we refer to these collectively as a MAMP or a PAMP. In the context of this class, MAMPs and PAMPs are the same thing. I usually use the term MAMP. So molecular associated, or sorry, pathogenic associated molecular proteins. So these are basically patterns that exist only on pathogens, patterns that only exist on microbes, so microbe-associated, pathogen-associated. And then we have DAMPs, so damage-associated. We may not even have time to talk about DAMPs, so we won't worry about that one in here. So these types of barcodes, so this is just a different type of barcode that can exist, is what we can also scan. So when our immune cells are going through, it'll scan our MHC, it says, hey, that's our cell, you don't have to worry about it. And then, oh no, we scanned a MAMP. This is not our cell, it's actually probably a pathogen. We need to do something about it. Here's our MHC. So in order to actually scan these, we ourselves, that being, actually have genetically coded receptors. Since these have been around for so long, as infected humans for so long, they have become a selective pressure on people. And our genes have mutated in ways that allow us to automatically detect these types of barcodes. All right, there's 3 different types that we have, at least that we're covering here. One's called toll-like. Toll-like exists on the outside of cells. So remember, extracellular versus intracellular. And then on the inside of cells, we have nod-like and rig-like. All right, so first let's talk about these toll-like. So here's our cell. These are those little toll receptors on the outside. So if any of these come into contact with a molecule or a barcode of what we know as a pathogen, it will start activating an inflammatory response or an immune response. But these are only good if the bacteria is outside of the cell. If the bacteria is inside of the cell, these aren't able to detect anything. So we essentially just have a copy of it on the inside of the cell called a nod-like receptor. All right, so nod-like and toll-like are basically the same thing. One's inside, one's outside. But we also have a third rig-like receptor that is specific for viruses. So when we come into contact with a MAMP, cells start signaling all the cells around them that something is wrong. And those happen with 2 major classes of chemicals. Cytokines is the first one. Cytokines includes lots and lots of different chemicals. There's only 2 specific ones that we'll learn later on. But cytokines, we're just going to include pretty much all the inflammatory chemicals. Just call them inflammatory signals. And then the other one is interferons. These terms may sound familiar to you, especially if you watch medical shows. And interferons are viral infections. All right, so we have scanned a barcode and decided that it's a pathogen of some kind. Now, we're going to signal the cells around us that something is happening. All right, so for cytokines, we're just activating the general immune system. Hey, this is a bacteria, something's going on. If it's a virus, we know that a virus is going to try to escape from that cell after it multiplies and get into other cells. So these interferons tell all the cells around it, they say, hey, I am infected with a virus right now. Make sure it doesn't get into your cells. So it will basically like downregulate lots of ways of getting in. So the endocytosis gets downregulated, and it's less likely to bring in that virus into new cells after it replicates and breaks out of the original infected cell. So these are really good when it comes to individual cells. Something is wrong with this cell, we need to get rid of it. What happens if a virus or if a bacteria, let's say, gets into a cell, bacteria or virus really, here's our cell, and something has gotten in and infected it, but it has masked itself, right? So not all of them will activate a toll or a nod-like. Let's say it found a way to bypass those other ones. Our cells can also change the appearance of the MHC. So they may not have to release lots of chemicals. There's this automatic response that if something gets in and starts messing around inside of the cell, It didn't trip the nod-like alarm or the rig-like alarm, but there are lots of other alarms that are going too. So we're just gonna change that MHC so it doesn't match our original barcode. Now, when it gets scanned, it'll say something is wrong here. All right, so let's indicate this as an X here. So it's changed this so it doesn't scan properly. When this happens, our body knows we need to destroy this cell before this thing can multiply and get out. All right? It has bypassed all our silent alarms, and now we need to mobilize a little bit better defense against it than we normally would. And when this happens, that's when we recruit this last cell type that we'll talk about, called natural killer cells. So natural killer cells, their whole job is go around and scan MHCs. That's all they do, all day, every day. They go around and they scan MHCs, and they look for something that's wrong. When they find something is wrong with an MHC, they kill that cell. It's very straightforward. Your immune system likes to kill things by poking holes in it over and over and over again until it dies. That's not even an exaggeration. Almost every component of your immune system, when it finds something it doesn't like, it stabs it to death. Chemically stabs it to death. It just pokes holes and then it eventually falls apart. So if we find something that looks weird, we kill it. And that's usually if there's something hiding away in there that's not making itself known. There's another way that this can happen, and that is through cancer. So cancer actually changes your MHC on your cells a lot of the time. And when a cancerous cell arises, one of these natural killer cells will say, hey, this is a cancerous cell, let's knock it out before it multiplies anymore. All right, so cancerous cells actually pop up in your body all the time. Like several per day is what's estimated the number of cancerous cells that occur in your body. But your natural killer cells are very good at sniffing them out. It's only when a cancerous cell develops that doesn't significantly alter the MHC that it's allowed to grow and multiply and become a cancerous state in a person. All right, so in identifying bad things and knowing who to eat and destroy, it's all about scanning these barcodes. Our MHC, see if anything's wrong, and using nod-like receptors, pole-like receptors, and rig-like receptors to find bacteria cells. And that creates a pretty comprehensive way of scanning through cells to determine what's bad and what's good and supposed to be there. But we also have other ways of protecting our tissues from pathogens. One being the complement system. So this is actually a really big, complex system that we're going to narrow down to pretty much one definition. Complement system is the guardian of your blood. It keeps things out of your blood. All right, there are a series of proteins. This says 20 proteins. It's actually more than that. This is an old slide. And the whole point of this is to keep bacteria, viruses, fungi, and eukaryotic pathogens out of your blood. What happens if a pathogen gets into your blood? It can go wherever it wants to go. So protecting your blood is protecting the highways from bacteria being able to get around and causing really bad systemic infections. There are actually 3 pathways. There's a lectin pathway too that I don't include in here. And so we're just going to call 2 pathways. We have a classical pathway. So if antibodies react, I know we haven't talked a lot about antibodies yet, they're on Thursday. And then there's the alternative pathway. So the difference is this happens if you run into, say, like a MAP or a PAMP, So all of this is detecting MAMPs and PAMPs and bacteria, so viruses, fungi, eukaryotic pathogens, and it will activate your complement system. Or if we have antibodies around, they're reacting with those. That's the really big difference between the two of these here. Once it does get activated, it uses what's known as a membrane attack complex. It stabs it until it dies, and then gets cleaned up by your white blood cells. Yeah? Poking holes is a really effective way to kill a bunch of cells. You don't have to know this diagram and how things work, but this is the cornerstone. It's called C3 protein, C3 reactive protein. I only show this because those of you who get into immunology sections of hospitals will see like a C-reactive protein test, and they're looking to see if something is in the blood. All right, so complement system protects us. Another systemic protection that we have is one that you are all familiar with, which is fevers. So fever is elevated body temperature, as we all know, are familiar with. A lot of people mistakenly believe that your body temperature rising by a few degrees is what kills the bacteria. But we all know now, 4 degrees is not going to stop an E. coli cell. It is a smidgen of its acceptable temperature tolerances. What this actually does is it makes it so that the iron in your body is not as easy to be taken up by the pathogens, which almost all need iron in them from our body. So we disrupt iron availability, and it prevents them from being able to multiply as much. With viruses and fungi, it can actually directly inhibit the replication, but we don't have to worry about those too much. What it also does is increase your metabolism. So as your temperature ramps up, metabolism ramps up, you're getting lots more enzymes being made, a lot more ATP being made, a lot more white blood cells being made, all of your systems working in overdrive to kind of eliminate that bacteria. So fever is not a bactericidal, like many people think it is. It's actually a bacteriostatic process, which helps your immune system catch up to that infection. And this basically happens through these little chemicals known as pyrogens. So a pyrogen is basically anything that causes a fever. They can be from bacteria or viruses themselves, or they can be a signal that our body produces, like these guys right here. All right, say, hey, we've detected a bacteria, we've detected a large-scale infection. Let's crank up the thermostat and try to drive these invaders out. So it activates hypothalamus. Not that this is a neurobio course, but this is what controls your body temperature. It also changes the constriction of your blood vessels to keep temperature in and not let it out through the capillaries near your skin. That covers fever. Fever is pretty straightforward. It's just these guys turn up the thermostat, and then keep in mind that it's a bacteriostatic elevation of metabolism that really drives that forward, helps clear out that infection. Any questions up to now? This is our last few slides that we're going to go over here. And I'm actually just going to use the whiteboard for this instead, so we can draw it. So inflammation is kind of the result of the activation of the immune system. All right, we kind of have our own idea of inflammation as something swelling up, which is what happens as a byproduct of inflammation, which is where we get our cardinal signs. This is the HERPA model. There's other acronyms that people use for this as well. Use whichever one you like. But this is usually what we think of with inflammation, especially like on a joint, on a cut, and you see it swelling up, getting warm, loss of mobility, and then hurting, possibly even forming a bruise. But those are in fact byproducts of what's happening in your immune system. So we're gonna go through all the different things that cause this to happen. So our example, like many that you'll find online, we're gonna say a splinter has broken the skin. So this is our splinter. This is skin. And let's see what's going on inside of the body here. Actually use these cell types in the proper areas. So on this splinter, there are microbes. They're everywhere. And there's two major signals that begin this. Let's mark that. Let me use this side for words. So first, we have tissue damage. So it's not just detecting a foreign pathogen here. Because if you think about like jamming your finger or breaking a bone, you get serious inflammation with that. But it's not because a bacteria was there, it's because something happened. It's tissue damage. So it was tissue that wasn't supposed to be there is now there. But that tissue damage itself actually releases lots of cytokines. And then you have bacteria, viruses, and ways of sensing that. So we have bacterial barcodes that are now present. So underneath our skin in this case, we have skin-associated lymphoid tissue. So at this point, we're beneath the dermal layers, we're in the interstitial layer, and our first big player is going to be our mast cell. Our mast cell has detected this stuff, and a mast cell is the war horn cell. This is the alarm system that happens underneath the skin for your immune system. So at this point, the mast cell starts dumping out lots of cytokines. Two is mast cell activation. And cytokine release. All right, so when a mast cell detects something and releases cytokines, this is a big step. This is saying, hey, there is something wrong. Activate everything, turn it on, and get it out of here. So these cytokines have various different purposes. We're going to talk about two main ones. The first is they will interact with all the capillaries at the area. So these cytokines will cause two major events to happen. Vasodilation. All right, so they'll cause those capillaries to expand. This allows more volume of blood through the area. And it also increases permeability of these. Increased permeability. Because all of those good white blood cells that we need to eat the bacteria, those are in your bloodstream, right? All those neutrophils circulate around in your bloodstream. So we want these neutrophils from here up to here, so that we can now ingest those bacteria and get rid of them. All right, so by increasing the volume of blood, you increase the amount of white blood cells in an area. By increasing the permeability, we can allow these white blood cells, these neutrophils, to squeeze out into this space. Right? And this is a process, we're going to say that, known as extravasation. All All right, so outside of the vessel, that's what extravasation refers to. And vasodilation and extravasation. Sometimes you will see extravasation as a more specific term called diapedesis. In our case, they mean the same thing. If you read diapedesis somewhere, it's referring to this process. Extravasation is actually a really broad term, but it's a lot easier to remember for most people. All right, so now we have neutrophils, and we have them in the right areas. So they start eating and phagocytizing all of the bacteria that exist in that area. We're trying to get rid of all of them. Now, this is an innate inflammation response. This is about the level of detail that you need to know. Not too bad. However, what we haven't talked about yet is the adaptive response. We'll do that on Thursday. And the major necessity for an adaptive response is information. So what is actually happening at the same time as all of this is we have a third cell type called an APC. APC cells. And these cells are essentially sampling a bunch of these bacteria, taking them prisoner, so to speak. And then they will travel, they will take that intelligence and relay it over. So we have heat from increased blood flow. We have swelling from increased blood flow, fluid accumulation, cell accumulation in interstitial areas. We have redness, because now there's more blood close to the surface of the skin. Altered function is just a result of all of this. What about pain? So there's actually a very special cytokine that's also produced. It's called prostaglandin. And prostaglandin stimulates nociceptors. Does anybody know what a nociceptor is? It's a pain cell. Nociceptors sense pain. So these prostaglandins that are produced by the mast cells, these are a type of cytokine that, and that contributes to the pain reaction. All right, so these are our 5 cardinal signs, and this is what's actually happening at the site. All right, and this doesn't just have to be the skin. This can be anywhere that inflammation happens at. Make sure we get silly. The last is what happens at the very end. So at the end, all the bacteria have been eaten. The neutrophils are actually dead at this point. They've eaten them all. They've released all of these toxic chemicals, and they've died. So the cleanup crew is the macrophages. So the very last step, macrophages will come in. They will clean up all the debris and the toxic chemicals here. And then they will make their exit through a lymphatic vessel. So your lymphatic vessels basically run alongside your circulatory system. So the macrophages will come in, clean everything up, go into a lymphatic vessel. It'll circulate through your lymph system, and then get dumped into your bloodstream, and then filtered out by your kidneys. And that's how you get rid of all of those bad byproducts after the process is over. The very last thing, before I let you go, is sometimes we can't get rid of whatever's in there like a splinter. And if you cannot get rid of it, you will still have this reaction over and over and over and over and over again. All right, asbestos that gets breathed in, that's one of the reasons why it's so dangerous. So what we often do, or our body often does, they create what's called a granuloma. You've probably heard the term granuloma before, where they wall off. So let's say the splinter got stuck. It would wall off this splinter with like scar tissue, essentially, and try to prevent this inflammation reaction from chaining over and over and over and over again. All right, questions real quick before I let you go?